Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture, we are going to talk about regulation of gene expression in eukaryotes. In the previous video lecture, we talked about regulation of gene expression in prokaryotes, which is very simple, as you can see. If you missed this video lecture, so go back to my channel and watch this video. Or I will give the link in the description box, then you will check it. Okay. In case of the prokaryotic, the regulation of gene expression is very simple, as you can see, but in case of eukaryotic cell, the regulation of gene expression is very complicated. So how the regulation of gene expression occur in case of the eukaryotic cell? So let's start it. Okay, friends, so let me write regulation of gene expression in eukaryotes. Now, regulation of gene expression in case of eukaryote, it is very complicated. Why? Because in this case, we have one promoter for one genes. But in case of prokaryote, we have one promoter for multiple genes. Means promoter is the region where the gene expression is controlled. Moreover, in case of eukaryotic, we have the DNA, which is a very complicated form. Means the DNA are wrapped around through a histone, which we call that nucleosome. So in nucleosome, the DNA are very complicated in form. And let's suppose in the nucleosome, what we have a particular area of the genes which need to be expressed, which need to be translate or transcribe and translate, which we get the protein. So it's very difficult and it's very complicated. Because at that particular point, first the DNA will be in a linear form, in a simple form. After the DNA are in a simple form, so then the protein will bind here. So it will cause the, it make a loop in a DNA. So when a loop is formed in a DNA, so then it will attract the RNA polymerase, and RNA polymerase will bind, and it will cause the production of one mRNA or one genes. One mRNA is produced, and then inside the RNA, again we have another process occur which we call it splicing. And in splicing, we have the intron portion, which is the non-coding portion, which is the junk portion, which should be removed. And after that, we get the exon portion, which are connected to each other, and we get the mature mRNA. And from this mature mRNA, we have the protein formed, but still this protein is the immature protein. And in this immature protein, we have actin and antine. The actin portion is actually used for the functional protein, and the antine portion is actually a junk portion of the protein. It should be removed. It is called as protein splicing. We already talked about the protein splicing in detail. We also talked the RNA splicing in detail. You can go back and watch it into my channel. Okay, so actually, whenever the regulation of gene expression occur in eukaryotes, so in this case it have a splicing process. All these processes are occur. So in prokaryotic, it's very simple. We have not splicing process. We have mature mRNA. From mature mRNA, we have protein, and then this protein will be used for different purposes. But in this case, what happened? We have first of all the DNA, which is a very in a compact form, and then it will be in a linear form, and then we get all the processes, which is again in a complex way. Okay, so let me explain that what happened in case of the eukaryotic regulation of expression in eukaryotic. Let's suppose this is DNA. Let's suppose this is a DNA, as you can see. So this DNA, this is actually the double-stranded DNA, and this DNA is actually wrapped around through a protein which we call that histone. Now this is actually called as what? This is called as nucleosome. Nucleosome. As you can see, this is a very complicated structure of the DNA. Let's suppose at that particular area, or let's suppose at that particular area, let's suppose the genes are present where we need a product or a protein for the cell. Okay, so it is very difficult to get protein directly. We cannot get. First of all, this will be, the DNA will be in a linear form. First, the DNA will be converted in a linear form, and then we get the protein and RNA and then protein. Okay, so for that purposes, what happen, there is certain type of the processes occur upon on the DNA, which we call it acetylation. So through this acetylation, what happen, the DNA will be, open. The DNA, the histone protein will be removed and the DNA will be open and the DNA will be in a simple form. Okay, then we have another process which we call that deacetylation. Okay, so in case of the deacetylation, what happen in case of the deacetylation, the histone protein are again binded and it will cause the coiling of the DNA, means the DNA will again form in this way, means the DNA are again in a nucleosome way, in a nucleosome structure. So deacetylation causing the cell to recoil, while acetylation cause the cell to be open. Then we have another process. This process occur by certain type of the protein and enzymes, different and multiple protein and enzyme. Moreover, in this case we have another process which we call that methylation. Now in methylation, both processes occur. Acetylation and deacetylation, both processes occur. In methylation, sometimes acetylation occur and sometimes demethylation occur. Okay, so this is deacetylation occur. Okay, so we have also another process, we call it methylation. But mostly acetylation and deacetylation occur. So when the cell are get ready for, or when the gene expression are regulated, so then acetylation will occur. When the gene expression are turned off, so then deacetylation will occur. When both processes are going on simultaneously, so then methylation will occur. Again, in these three processes, the protein and enzymes are very necessary. Multiple protein and multiple enzymes are needed in these processes. What happen? Let's suppose if we get the DNA in a linear form, as you can see, DNA, let's suppose this is a DNA in a linear form. Mostly DNA are in a double stranded. So then what happen? We have one particular area which we call that promoter. And this promoter can control one gene. Let's suppose this gene is A. It can control one gene which we call that A. Still in this case we have at upstream. I hope you guys know about upstream and downstream of the DNA. Let me a little bit tell you what is upstream and downstream. When the DNA is 5' to 3', at 5' to 3', the sequences are called as downstream. Then from 3' to 5', the sequences are called as upstream. Okay, so this is the promoter. So at the promoter, after the promoter region, means at 5' to 3' of the promoter, we call that downstream sequences. And behind the promoter region, or from 3' to 5', we call that upstream sequences. Okay, now what happened at upstream of the promoter region, we have some other sequences which we call that enhancer. So enhancer is actually the sequences where the transcriptional factor are binded and it can cause the gene to be expressed. Okay, so let's suppose this is the enhancer portion. So at that portion there are some sequences where the transcriptional factor are binded. Transcriptional factor, let me a little bit write. Transcriptional factor is nothing but these are what? These are the activator proteins, and we have inhibitor proteins or repressor protein, inhibitor or repressor as well. Okay, now there are some sequences which we call it enhancer sequences. At the enhancer sequences, the activator protein are binded. So when the activator protein are binded to the enhancer sequences, so then it causes the DNA to be bent. So let's suppose this is the DNA, as you can see. So then when the protein are binded, so then it will be converted into this way, as you can see. So let's suppose again this is the promoter region. So this will be the enhancer, and inside this enhancer what we have the transcriptional factor, which is actually the activator. This transcriptional factor will cause the attraction of the RNA protein when the loop is formed. So then it will cause the attraction of what? RNA polymerase. So then RNA polymerase will attract and it will bind. Let's suppose this is the RNA polymerase. RNA polymerase bind here and it will cause the gene expression, or we get what? We get the RNA. Okay, this is how the process will be activated. So when the polymerase enzyme are going on, so then it will transcribing the RNA, which we call that mRNA. But again, as I told you that it is very complicated. We get RNA also, but still this RNA is not the mature RNA. And said this mRNA, we have what? We have the portion which we call that exon and intron. Now let's suppose these are the portion, as you can see. These green are the portion, as you can see. These are what? These are the exon portion. Exon, okay? E means exon. And there are some reason which the cell do not need. This portion is called that intron. I. Intron is actually the junk portion of the RNA. It is that sequences from which the protein are not formed. This is the junk portion, and it should be cleaved out or removed out from this RNA. So when it is removed out, so then we get a protein. Okay, so it will be removed by a process we call that splicing, which we already explained in previous videos lecture. You can watch it into my channel, Splicing. So through splicing, the intron portion will be removed and the exon portion are connected to each other. So when the exon portion, let's suppose this is the exon portion, are connected to each other, we call that mature mRNA. So then we get the mature mRNA. So when we get the mature mRNA, so then it will convert it into a protein. So we get what? We get a protein. Again, when we get the protein, so remember guys, in case of the protein, let's suppose this is the protein. So again, in case of the protein, this protein is not a functional protein. So what happened? This protein need to be functional, which we call a post-translation process. So in this process, what happened? Again, the splicing will occur, which we call that protein splicing. So in protein splicing, what actually happened inside the protein? Again, we have the portion which is the junk portion and which is the important portion. Let's suppose these green portion are. The portion which we call that actin. So these are the most important portion of the protein, which are actually the functional part of the protein, and it is needed. And then there are some portion which is actually I have drawn with a blue marker, which actually I am drawing with a blue marker. This is actually actin. This portion should be removed from the protein. We call that splicing. So when the actin portion is removed and actin portion are joined together, we call that protein splicing. So then the protein is matured and it will perform their function. As you can see, the process is very complicated. So means that in this case, the regulation of the gene expression occur at different level. It will be regulated at that level. It will be regulated at that level, means it will also regulated at the that level, means at transcription level and at post-transcription level. Let me once again tell you that regulation of the eukaryotic, in case of regulation of gene expression in eukaryotic, the genes will be regulated at different level. It will be regulated at this coiling and recoiling, means this is transcription of the DNA portion. It will be regulated at the translation of the RNA, and it will also regulated at the post-translational of the RNA as well. Okay, when we get the protein, so then this protein is used by the cell. Let's suppose a lot of proteins are formed. Let's suppose, so then this genes, let's suppose this is the genes which is actually transcribing. These genes need to be stopped. So then at that specific portion, upstream or downstream. At the downstream, let's suppose there is a sequences which we call it silencer or terminator sequences. Okay, so what happen? Inhibitory protein are activated and it will bind to this silencer portion or terminator portion. So when it is bind on this portion, let's suppose these are the inhibitory protein. When it bind, so then RNA polymerase cannot passes, and when RNA polymerase are not passes, it means it is actually present at that portion, at upstream. Silencer sequence is also present upstream and downstream as well. But let's suppose here I am drawing the silencer sequences at that portion. So when the protein which is called it inhibitor or repressor protein are bind, so then the RNA protein are not binded, because already the inhibitor protein are binded and it occupy the place of the RNA polymerase. If it is occupying the place of the RNA polymerase, so there is no particular area where the RNA polymerase is binded. So when the RNA polymerase is not binding, so the gene expression will be stopped or turned off. So this is how the gene will be turned off. When the gene is turned off, so then it is again converted into this form, into nucleosome. Again, the histone protein are binded and it will cause the coiling of the DNA. Deacetylation process is occur. So through this deacetylation process, the process will be stopped, and when the process stopped, so then the gene regulation will be not occur, post-transcription, all these things are not going on. So this is how the regulation of gene expression occur. Okay, so through this process we can stop the regulation of gene expression. We can turn off the gene regulation inside the eukaryotic cell. So that's all about the regulation of expression in eukaryotes. I hope you guys get an idea about that. If you like this video, make sure to hit the like button, share this video, and definitely subscribe my channel to get more interesting videos like that. Inshallah, in the next video we are going to explain operon concept in detail. Thank you so much for watching that video.